Okay, everyone, welcome to another episode of Raising the Bar with myself, John Cooper. And today I'm very lucky and very grateful to have on the show Dr. Thomas Cowan. Dr. Tom Cowan graduated from Michigan State University College of Human Medicine in 1984. After his residency and family practice at Johnson City Hospital in New York, he set up a medical practice in Peterborough, New Hampshire, and served as Vice President of the Physicians Association for Medicine, and is a founding board member of the Weston A. Price Foundation. During his career, he has studied and written about many subjects in medicine. These include nutrition, medicine, and herbal medicine. He is the author of numerous books and will be talking about his book, Contagion Myth, which he co-wrote with Sally Fallon Morell. Tom, welcome to the show.
Thank you. Thank you for having me on today.
Would you like to comment on what's happened to you, and a bit of a backstory and how it led you to the book and obviously the response you've had from it, negative and positive?
I mean, it's not, I wouldn't say it's a particularly exciting story, except, I happen to have grown up in a world of very high-powered doctors and, people. And I didn't really think they knew a lot. And I don't know why, I just didn't. And so I just went into it and this thing, and I had a hard time getting myself to believe things I didn't believe. There wasn't any particular revelation. I just didn't follow the logic or didn't follow the thinking. And then I started hearing other ways of thinking about things. I heard the heart isn't a pump. Everybody said the heart is a pump. So I spent 20 years looking into that and decided it's true. The heart is not a pump. Blocked arteries don't cause heart attacks. Vaccines are not safe and effective. And cancer is not a genetic disease by any stretch. And, I just kept going forward. I knew that viruses didn't cause disease for probably 30 years, but I didn't really want to get into that because I thought it was too complicated, although it turns out it isn't. And then I sort of stuck my foot into it inadvertently, and 100 million people heard me say that viruses don't cause disease. So then, being the normal person that I am, I decided I should look into it and figure out whether I was right or not. Because the other thing I found out is in all the writings and books, and I don't know how many lectures I've given, it turns out I got a lot of things wrong. And here's the interesting thing about that. I can say, I think categorically, that every single thing I got wrong because I believed what I had been told without questioning.
Yeah.
And a lot of those times I would come to a fork in the road, and there's a saying, you come to a fork in the road and take it. So I took the road that seemed easiest at the time. Like, well, I don't really want to get into this, so I'll just believe this right now. An example would be the so-called lipid bilayer membrane. Everybody thinks that cells have this membrane around them that's made of two layers. Well, it's like a sandwich of 2 layers of protein and the lipids in the middle. And it turns out there's no picture of that in any published scientific literature. There's really no evidence that that's actually true. It's an elegant theory, but doesn't happen to correspond to reality. So that's just an example. And, the next step for me was really doing the work to find out whether whether I'm right or not. And so I ended up having to understand what we mean by the question, does a virus cause disease? And so I think I have a way of explaining that. If you're interested, I can go through it.
Yeah, I would love to.
And honestly, my goal here is I don't want anybody to believe me. I want people to understand this for themselves so that they can then ask whoever they want, including virologists. And you see, once you understand it, and I can tell you, I can guarantee even from knowing you from 5 minutes that in a half an hour, you will understand everything you need to know about this. So nobody will be able to do this to you again, because you will see it as clear as a bell.
I love that.
There are some, complications, which, but more or less, so here's, and so I'm going to do this step by step. And I just encourage you, if you don't get or understand or agree with any of the steps, you should stop me and then we work on that step.
Yeah.
Right.
Thank you. Yeah.
So you're not believing me. You're not guessing. You're not doing anything except I get exactly what he's saying.
Thanks, Tom.
Okay.
Yeah.
So those are the ground rules. Now, the first thing is, we're talking about, the subject here is, does a virus, and in this case, we're talking about, does a new virus, meaning the, SARS-CoV-2 virus, cause a disease called COVID-19? Now, COVID-19 is itself problematic because there is no actual definition of the disease. But let's skip that for a minute. And because it could be chickenpox, and that's very characteristic. It could be measles, that's very characteristic. So let's say there is a well-known or recognized illness called COVID-19. So now the question is, does this virus, which we're told causes it, I mean, everything that we're doing, it comes from that understanding, right?
Yeah.
So that's the question. So the first question I would ask you is, do you agree that the virus is a thing? And specifically, I mean, in contrast to an idea or an emotion or a philosophical position in life. In other words, it's a thing, like this pen, that's a thing.
Yeah. Yeah.
You agree with that?
Well, I'm meant to believe that, yeah.
Right. That's a complicated question a little bit, but everybody who thinks the virus causes the disease thinks it's a thing that you can measure and characterize and, know what it looks like. There's, computer pictures of it, etc. Right? So we're talking about a thing.
Yeah.
Now, the next thing I would say is, when you ask most people, why do you think, or how do you know that the virus causes disease? They'll say, well, a lot of people got sick in Wuhan, and then it spread to Italy, and then it went to England or China or Russia, or my Aunt Bessie went to a party and then she got sick, or people weren't sick at San Quentin and then somebody went there and then they all got sick. And the fact of the matter is, those are epidemiological observations. In other words, they're things that epidemiologists come up with to generate hypotheses that they can then test to see what the cause is.
Can I just ask, what is the definition, or can you give us the definition of an epidemiologist, just so we don't skip over that?
They're the ones who say a lot of people get sick and we need to find out why.
Right, okay, thank you.
And nobody in science thinks that's how you prove causation. That's how you generate hypotheses so that you can test. And so the idea that there was something contagious that we can't see, let's just say I agree with. And so we should test that hypothesis. I don't need more examples of Uncle Fred going to a party and getting sick. And the reason for that is, you cannot say, because a lot of people get sick in the same place, that means it's a virus, unless you think that Hiroshima was a virus. Or you can't say because something spreads from one place to another, it's a virus, unless you think Chernobyl was a virus, because that spread all over Eastern Europe.
It's a correlation, not a causality, basically. Yeah.
It's an observation that is meant to generate studies to see whether this is the cause.
Yeah.
And we have some great examples, like for 100 years, people thought scurvy was an infectious disease, because one sailor after another, their teeth fell out and they went into heart failure and died. And then they would go to a shore, and then the next boat would get the same thing. And so they thought it was something spread between people. And then it turns out somebody ate a lime and the whole thing went away. And so there are consequences of being wrong. So we need to be correct about this. And I don't need any more epidemiological observations to get me to think we should look into whether it's caused by a virus, right? So we got that. So now we're going to look into it. Now, interestingly, most people at that point, when you say, how do you know if a virus causes a disease? Because you just told me you were 100% sure it does, and they have no idea. And which is interesting, because if you have no idea, not just whether it's been done, but how you would do it, that's a problem. So here's how you do it. The first step in this is you have to isolate the virus. Now, what do I mean by that? Let's take another example so you can see. And this is where I'm going to ask you questions. And so please pay attention to the words that I use here.
Sure.
Our quest is to find out whether caffeine found in coffee beans causes high blood pressure. Got it?
Mm-hmm. Yeah.
So I take coffee beans, I grind them up in a coffee grinder, and I put them in a capsule, and I give 10 people ground up coffee beans and 10 people something that's a proper placebo. So I do proper controls. All 10 people I give the ground coffee beans get high blood pressure. None of the placebo do. Have I proven that the caffeine in coffee beans causes high blood pressure?
Not to me, no.
Because why?
Because it's a correlation, not a causality, because there might be many other elements to that coffee bean which contains poisons and toxins that could also be resulting in the atrophy of these people. So to reduce it down to just caffeine is it's like someone eating a 10-course banquet meal and blaming it and dying of poison and blaming it on the salt content.
Right. I would forget about the correlation and causation part of that.
Right.
I think what you mean, correct me if I'm wrong, is we haven't isolated the caffeine part.
Exactly.
In other words, there's fiber and caffeic acids and a whole lot of other things. So you have no idea whether it's the caffeine or not. Okay. So let's do another step. We take the ground coffee and we put it through a filter paper, and then we get coffee, and then we throw away the fiber part, and then we do a control. We give that coffee to 10 people. 10 people, we give them a similar liquid that's just water and a food coloring or something. The 10 people with the coffee get high blood pressure. The 10 people who get the placebo don't. Have we now proven that the caffeine in the coffee beans causes high blood pressure?
Can you tell me about the filtering system? What exactly was the filtering system?
Anything that's not soluble. So like it's fiber and anything that's water soluble will go through the filter.
Well, I'm guessing toxins could still be passed through. So I would say it's not totally isolating the...
Right. Yeah. In other words, you haven't isolated the caffeine. You now have less things, but you still have caffeic acid and oils and a whole bunch of other things.
Yes, it's not a pure isolation, I would say.
Right. So then we do a next step, which is we take the coffee and we put it through some gradient and a density gradient. We centrifuge it, and then it spins things out by weight. And then we have one band that we suck that out, and we send it to a laboratory, and they confirm that the only thing you have in that sample is pure caffeine and nothing else. And then you send it to another lab. They do the same steps that you did, and they say, yes, this is only caffeine. That's what you have. You give this to 10 people. 10 people, you give them a control that looks the same, but it's nothing. They get high blood pressure. Have you now proven that it's caffeine from coffee beans that causes high blood pressure?
Not to me.
Because
there's many other factors that that can affect disease. So it's...
Yeah, I disagree. That's pretty damn close.
Oh, is it? Okay.
Yeah, I mean, you've proven and had independent people show you that the only thing you have there is caffeine.
But what if the people that they tested on, they had underlying health issues, or they were obese?
Yeah, they're a proper control.
Okay, so it's properly controlled, so that all the people that stay healthy and they're all of the same stock and
Whatever. They're the same as the other group.
right. Okay, so I mean that's very hard to reduce everything down to that level, I would say. But if that's the case, then I would say there's a strong influence of that caffeine causing the heart issues or whatever the disease was. Yeah,
Yes, I would say that's
yeah.
good enough.
Yeah, it's good enough. Yeah, I would say so. Yeah, I'm actually
Now,
I'm trying to think so outside the box. Maybe I'm too outside the
yeah,
box.
no, you don't have to think so outside.
Yeah,
Just,
okay,
just
yeah.
think normal.
Okay, yeah.
You want to know which things draw a line. You got two things. You want to
Yeah.
know whether it's a yellow or a blue pen.
Yeah.
You separate it out. You only have a yellow pen.
Yeah. Yeah.
And then you see if it draws a yellow line. You know that this draws the yellow line. That's how human beings think,
Yeah.
right?
Yeah.
Now, what if after the coffee stage, you had mixed the coffee with a vat that contains tea and yerba mate and chocolate and 10 other caffeine containing liquids, and then you did the centrifuge and pulled out the caffeine, and then you gave it to people, would you still know that it was the caffeine from coffee beans that caused high blood pressure?
Only if the centrifuge was able to create a pure isolation, but I'm not. Does it or not?
So there's no way you can separate the caffeine from the coffee beans from the caffeine from chocolate.
With the centrifuge.
Yes, with anything.
Yeah, then you wouldn't say that is the result of caffeine. You would say that could also be as the result of all the other ingredients.
No, not true. You could say it was from caffeine, but you don't know that the caffeine came from the coffee.
Oh, yeah, of course. Yeah, because tea has caffeine. Yeah, sorry. Yeah, I wasn't thinking there.
Right. You know it's caffeine.
Right. Yeah.
But you don't know whether you don't know where the caffeine came from.
Sure. Yeah, because it could come from the teas. Yeah. Okay.
Right.
Yeah. Yeah.
So you could say it's caffeine, but not from coffee.
Exactly.
Now, here's the point. That is how a human being does isolation and proves causation. Now, it's the exact same thing with viruses. You take somebody who's sick, right? COVID-19 positive test. You take some of their snot. That's where we say the virus is found. You macerate it. That's the coffee grinder, right? You put it through a filter paper. You get rid of the fungus and the bacteria and the dead cells and all the rest of it. You've got a liquid. That's not pure virus, right? That's a whole lot of things. Then you centrifuge it in a density grade. It comes out in a certain band. You take the band out, you send it to a lab, or you do it yourself. You have somebody check you, do all these steps. You say, the only thing I have there is virus. You show a picture of it. The only thing I have there is virus. You give that to an animal. They all get sick in the same way as the original person. You've now proven that the virus that you've isolated causes disease. Once you've isolated it like that, then you can take out the DNA or the RNA, whichever it has. You can characterize it and say, here's the sequence. And then, and only then, could you do a test to show that this has a unique piece of RNA or DNA that no other thing has. But unless you've isolated it, you can't possibly say that. Right?
Yeah. What they're calling isolation isn't isolation. It's actually just jumbling it up with lots of other things.
I'll tell you exactly how they do it. Yeah. So that process that I just described, snot, macerate, filter, centrifuge, prove that that's all you have, was done for 20 years between the 30s and the 50s. And not once were they able to make an animal sick. Not once. They proved that viruses isolated like this are not pathogenic. And so that was the end of it. And except then somebody, a guy named Ember, said, let's try it a different way. And he said, okay, I take snot for measles secretions and I grind it up, just like I'm supposed to. I filter it. And then I put that on kidney tissue cells. So that's not purified, right? We already know that. and then I poison the kidney cells and I starve them, and then they break down into a million pieces. That's like putting the coffee in a vat of tea and chocolate, etc. And then for some God knows what reason, I claim that proves that that virus causes disease. It's complete logical nonsense. And that's what they're doing. Every study that has said they isolated this virus has done the exact steps that I just said. They even now, they don't even take somebody who's sick. They take cultured stuff that has not ever been isolated or purified. They filter it. They inoculate that on kidney tissue. That doesn't grow, so they starve and poison it. It breaks down into a million genetic pieces. Then they do a genetic analysis of those pieces and say they found a virus.
Oh dear, oh dear.
And that's nuts. Now, the point I'm making here is, first of all, I think you now know why that's nuts. Because that's not how any human being isolates one thing from any other thing.
Exactly.
Here's the point that a lot of people don't know. This is not technically difficult. Nobody has said, here's a person who's sick, and I macerate it, filter it, centrifuge it. Here's this virus. And there's not one study in the medical literature that has done that. It's not hard. They did it for 20 years. They can't, the reason they don't do it, they say, is because they can't find the virus like that. Which, you're right.
Yeah, right. That's the truth. And that's the truth, yeah.
Yeah, you can't find it because it's not there, not enough of it there. Then how in the heck do you think it's causing everybody to die? If you can't even find it using an electron microscope where you can find like one, then, this is nuts. So then the problem is the kidney's tissue has the same genetic material as the so-called virus. That's like putting it in a vat of other things that contain caffeine. So from then on, you have no idea where this stuff came from. And that's where we are. And because of that, the tests for the genetic material are not specific to any virus, right? Because they don't know where it's coming from. And not only that, the people admit it. When the Christian Drosten paper that started this, and he gave the sequences that you're supposed to look for to find the virus, and he says, and I can read his quote, hang on. Quote, this is from the original paper that started this. We aim to develop and deploy robust diagnostic methodology for use in public health laboratories without having virus material available. That's like, the analogy here is, I say to you, I want you to build an exact replica of King Beauregard's castle in England from 1226 out of Lego. And here's all the Lego pieces of the world. Okay, ready, set, go. Do you have any questions for me?
You give me all the Lego pieces and you want me to recreate a famous building?
The exact replica of his castle.
Oh, well, not the, no, not, of course, not the exact. Only a Lego recreation, yeah.
No, but an exact Lego recreation of it.
No, it's not possible.
Maybe, but would you like to see a picture of the castle?
Yeah, you'd think you'd need to, yeah.
Right, you can't.
No.
So you have to make the exact replica, but I'm not going to show you a picture of it. And you would say to me, that's not possible. But I'm going to give you $1 million, so you give it a try. So here's what you do. You look through the Lego pieces and you find a turret and a moat and a flag. And you say to yourself, these must be part of castles. And then you put it into a computer and it recreates the entire castle out of these few pieces.
Right.
And then you say that's the castle.
Mm.
And then you give it to somebody else and he does the same thing and his computer makes a slightly different version, right?
Mm. Mm.
Because there is no castle.
Mhm.
And he makes a different computer version. And that's why we have 397,000 different mutations of the coronavirus.
Mm.
Because everybody's making their own castle. 50 years later, the original guy who gave you the challenge said, oh, by the way, King Beauregard, he was afraid of snakes, so he lived in a townhouse in London. He didn't have a castle. That's where we're at.
That's where we're at.
And what they say, the technical word they use, what is the virus? It's called an in silico virus. And that means a theoretical virus in a computer. Mm. And so they just recreate an in silico genome of an in silico virus using their computer program in a process called alignment. Mm. And it's completely imaginary.
Yeah, so when they're looking through a microscope, what are they actually looking at when they're showing us pictures of these things?
So most of the pictures are computer generated models.
Right.
Right? Now they show you pictures of particles but those are particles that you don't know where they came from because they were from snot that was grown on kidney cells, which then we know that when tissue breaks down because you starve and poison it, it makes these little particles and they're sometimes called exosomes or cellular debris. Yeah. And they have no pathogenic effect. And that's what they're showing you. Right. But by the way, here's another thing. If I was to say to you, I want to show you what your hand looks like so we can figure out what your hand does, right? Yeah. That's our quest. So here's how I'm going to do it. I'm going to take your hand, I'm going to cut it off your arm, and then I'm going to put it in an enzyme vat to dissolve all the tissue, and then I'm going to freeze it at minus 200 degrees, and then I'm going to bombard it with electrons, and then I'm going to cut it into little pieces, and then I'm going to put a bunch of metallic stains on it, and then I'm going to put it under a microscope, and then I'm going to tell you what your hand does. You think that's a reasonable approach?
It's a bit messy, I have to say.
That's what they did to find the virus. They took somebody's snot and they essentially froze it, poisoned it, and every picture that you see on an electron microscope is not a picture of the thing. It's a picture of heavy metal stains on something. And nobody has any clue whether it has any relation to what was found inside a living tissue. These are artifacts, and they're artifacts of dead and dying tissue. When you poison somebody with arsenic, air pollution, fear, electromagnetic fields, their tissue breaks down. They make all these particles that have genetic pieces in it. And that's what they're calling the virus.
Right. And what's the actual name that we give that, or you would give that? The things that they're looking at, that's not exosomes, is it?
All anybody really knows is that when you starve and poison a tissue, whether you do it in a so-called viral culture or whether you do it to a real person, their tissues break down into these little pieces. Some of them are encapsulated, some of them stain. Yeah. They have all different names, but they're not pathogens from the outside. And here's another quote, and sometimes they're called exosomes, but I would even caution that. Here's an article from Viruses July 2020. And so this is the premier journal in the world for viruses. And they were saying the difficulty of separating viruses from exosomes. In other words, they don't know whether this is coming from the outside or something generated from inside. And their final statement was, and I'm quoting, "To date, a reliable method that can guarantee a separation of exosomes from viruses doesn't exist." Now, here's the way to understand this. I have 2 forks and a knife, right? I can separate the fork from the knife, right? That's called isolation. And that's why I said a fork is a thing, like a virus is a thing, or at least they tell us it's a thing.
Yeah.
Now, if I cannot separate this fork from this fork, do you know why?
Go on.
Because they're both forks. And the only reason you can't separate one thing from another is because they're the same thing.
Right.
And so we used to think that these viruses were infections from the outside. And we now know that you can't separate them from breakdown debris from the inside. And there's no way to separate them. And in 20 years, everybody will know that all these things we call viruses, all these things going back from smallpox and measles, which, by the way, they had a German Supreme Court case where they ruled that nobody has proven the existence of a measles virus.
Yeah, Stefan Lanka, wasn't it? Dr. Stefan Lanka.
So now they'll know that all these things are not from the outside. They're not making us sick. There's no evidence of that. They're simply breakdown products found by the way that electron microscopes create artifacts, because they have to macerate, freeze, bombard with electrons, poison with heavy metals in order to see anything. And here's another thing. So here's how they say this virus makes us sick, right? I'm going to admit in a minute that I'm going to embellish the description, right? So this is a artist's rendering of the description, right? So you have this virus that has a erect spike protein protruding from its surface. This erect spike protein finds a receptacle, a receiver, a receptor on the soft cell membrane of the cell. It forces itself into the receptacle, right? The receptor called the ACE2 receptor, and injects itself into the cell, has its way with the cell, takes over the cellular machinery. The cell then is spent and destroyed, and the virus emerges victorious from the cell to find its next victim. I mean, I don't know about you, but that sounds pretty weird to me. But here's the interesting part of that. Do you know how many published pictures there are of a spike protein attaching to a ACE2 receptor in a cell membrane?
I don't know. I'm guessing not many. 0.
The exact answer is 0. In fact, do you know how many pictures there are in the published literature of an ACE2 receptor in a cell membrane?
That language doesn't really make sense to me, but I'm guessing not many. I'm guessing from what you're saying, none.
None. So what that means is, they say that the way the virus causes disease is it has this spike protein, right?
Yeah.
That attaches to a receptor on the cell, right?
But are we talking about you to me, like from person to person? Or are we talking just we're just taking one person's body here?
No, from one person.
So let's say you transmit this to me, and then it goes into my cell membrane. Yeah,
so now you have this virus inside you.
Yes. Okay.
How does it cause disease? The spike protein attaches to your cell membrane through this so-called ACE2 receptor.
Yeah.
And then injects itself into your cell. This is a very sort of warlike, like sexual domination image they're using. And my point is, and then it kills your cell, and then it emerges and goes find somebody else. The point I'm making is not one part of that process has ever been documented.
Not one.
Not one. Now, they have circumstantial evidence that it's the case, and I don't want to get into that evidence right now, but that should be shocking to everybody. And it's the same reason why we have, 17 different models of the cell membrane versus one model of what a schnauzer looks like. The reason we have one model of what a schnauzer looks like is the dog. Is you can go and find it and say, that's what it looks like. With a cell membrane, because nobody has actually seen it, they have all these elaborate models, which nobody has ever proven that any of them have anything to do with reality. And yet, that's what we're basing this whole exercise on, these models of reality, which turn out, if you actually do the science, are completely unproven, unprovable, irrational, and nonsensical. And that's why, for me, it's so important to go through this, because if we just say, well, it's just a bad respiratory virus, and so they'll just do it again and again and again, unless we understand that the whole thing is just a scientific construct, which has no basis in reality. We've been down this road with AIDS and Zika and Ebola and MERS and SARS-1, and they keep rolling out the same playbook over and over again. So it's about time some of us know whether or not this is true. And that was a long rant, but
no, God bless you. God bless you for speaking out, speaking up. So we're saying that there's nothing transmissible then, nothing, and nothing, no pathogen that's.
There's
what about bacteria though? What's it? Bacteria and viruses. I mean, if you touch something dirty and ingest that, that is effectively like poison, isn't it? That like you would become sick from that.
So here's the thing. So that's the virus story. What about the bacteria story?
Yeah.
So, it's the same thing. If you want to tell me that that the bacteria cause disease, obviously you have to isolate the bacteria, right? Mm-hmm. You cannot say, I know this thing causes anything, unless that's the only thing you have. So even take the famous example of Semmelweis, right? He's the guy who said, you got to wash your hands after you do autopsies because you're going to make women sick if you deliver their baby, right? That's how this. So here's a bunch of doctors who go from an autopsy of somebody who obviously just died. They're embalming them with formaldehyde, one of the worst poisons there is, and they have all this like dead tissue and formaldehyde on their hands, and then they go stick their hand in some woman's open cervix who's giving baby, and the woman gets sick, right? Now, how do you know that it was only the bacteria in there that got the woman sick?
Yeah.
Do you know that? No.
Yeah, we don't know that for the reasons you said earlier.
Now, some people say, so do you mean that, am I saying people shouldn't wash their hands? No, because I don't think you should stick dead human diseased parts and formaldehyde in a woman's open cervix who's giving birth. Now, if you want to convince me that bacteria, it's only the bacteria, you have to isolate the bacteria. Now, here's another thing. If you can show me isolated bacteria make anybody sick, I will retract the book. All you have to do is isolate them and give it to a person and make them sick, or animals, and I will say I'm wrong. So here's what happens. Let's take a common example. So somebody has so-called strep throat, right? So we know that if you isolate the strep and expose another person to it, they don't get sick. Everybody has strep. So here is, in fact, what happens. Something happens to you, like you eat a whole bunch of sugar, or you put metal in your mouth and that attracts electromagnetic fields, and then you get diseased tonsils. The tonsils start dying. It's the same thing that happens, in a compost pile. If you put dead squirrels in your compost pile, next thing you know, you'll have bacteria come to eat the dead squirrels. Nobody thinks, A, that the compost pile has an infection. And not only that, if you take the bacteria from that pile and put them into a healthy compost, they don't grow. The role of bacteria in nature is to digest that which is dead and dying. That's their role. So you have dead and dying tonsils because you eat crap, because you have, metal in your mouth, because you're using fluoride, which poisoning your tissue. And the bacteria come to help you out and digest the dying tissue so you can start over again. They're your best friends. Now, it is true that if you kill the bacteria with like antibiotics, that will stop the process. And the only thing that'll happen is either you'll do it again or you'll get even sicker. And next thing you know, you've got heart disease. Because that resurrection process, that rejuvenation process, you thwarted because you don't understand the problem. It's exactly like what I've said for decades. If you get a splinter in your finger, you make pus to get the splinter out. Is the pus, that's an infection. Is that the disease?
No, it's just a reaction.
The splinter is the disease, right?
Yeah.
The pus is the therapy for the splinter.
Yeah.
What about if you smoke and put debris in your lungs? Then you get mucus and pus to get the debris out of your lungs. Is that an infection or is that a resurrection process?
Yeah, the resurrection process, healing process.
So you can 100% predict that if you're dumb enough to think that like doctors, that the pus is the disease and you stop the pus, A, the person will do it over and over again, or they'll build up a pocket of debris in their lungs, which we then call lung cancer. And then we say, how did you get lung cancer? Well, you must have smoked. And somebody stopped you from getting the smoke out of your lungs. And I would submit that not only is that the way we get smoke out of our lungs, it's the only way. We don't have a backup plan. You get debris, you poison your tissues, your body creates these bacteria, they go to the place, they digest the debris, they get rid of it, and then you're good again. There's no other backup plan. And if we don't understand that, we'll end up with a whole world of chronically sick people, which is what we have.
Yes, what we have.
So Tom, what about people that say, I know people that have had it. I know people that have had, COVID, let's say. What's actually happening for that? What's actually the reality there then?
First of all, stick with, I sometimes will say to people like that, so we've already agreed that it's not a virus, right?
Right.
And if they say no, I'll go back over the whole thing. Here's how you find a virus. Here's how you prove it caused. We did that. We couldn't find a virus. We can't sequence the genome. We can't prove it caused. There's no published study. There's nothing. If you don't agree with that, then there's nothing to talk about.
And if they say it has been sequenced and they reference certain, you just say that's not proper sequencing, that's culturing.
Then you ask to see the study. And every single one I've seen now, 32 different studies that people have sent me. I've seen people claim it was sequenced and isolated 125,000 times. I don't know if that's the exact number. I've looked at literally hundreds of those. Not one is the isolation sequencing that I just talked about.
Yeah.
They don't do it.
I also had a guest on who did a freedom of information request to all the major medical institutions, and they all came back and we said we cannot provide the information that states that there is a pure isolation, only with the culturing process of monkey cells they put in, I think it is.
Yes.
Yeah.
So that's what I'm trying to get people like you. Now you know what to ask.
Yeah.
And then you could ask them, why don't you just take somebody with snot, macerate it, filter it, centrifuge it, look at it under a microscope, see what you got.
Yeah.
That's how they did it for 20 years.
They'll say that they'll think that is what's been happening, I think.
They do, but they don't, but they haven't. Then you say, let me see the study. There isn't one.
Yeah.
The same with measles. There wasn't one, and the Supreme Court ruled This is nonsense. This is just broken down kidney cells.
Yeah.
It's funny because Stefan Lanka, he originally, put the 100,000 bet up for anyone that could prove that measles existed. Initially, the court ruled him straight out, thinking it was just baloney, and then he appealed it, and then he won the case. Didn't he? I think people need to know that.
Wir stehen hier vor dem Oberlandesgericht Stuttgart und hier wird heute verhandelt, ob es das Masernvirus gibt. Im Jahre 2011 hat ein Biologe namens Stefan Lanka ein Preisausschreiben veröffentlicht, in dem er 100.000 € versprochen hat demjenigen, der ihm beweist, dass es das Masernvirus gibt. Dr. Stefan Lanka, das Urteil ist gesprochen und Sie lächeln. Warum?
Weil ich dieses Urteil erwartet habe. Mir war klar, dass das, oder ich habe vermutet, dass das Gericht nicht in die wissenschaftliche Beweisaufnahme geht und dass es an der Formalie scheitern wird, nämlich so, wie ich es gefordert habe. Ja? Und das ist eindeutig, daran gibt es nichts zu rütteln. Ein zweites Statement dazu, eben das Gericht hat das festgestellt, dass innerhalb 6 Publikationen kein Virus nachgewiesen worden ist. Aber es sind ja alle Zitate zu berücksichtigen, ja, und insgesamt musste ich ja 3.360 Publikationen berücksichtigen. Das war eine Höllenarbeit, riesen Rechercheaufwand, ja. Aber die Aussage des Gerichts ist ganz klar, in 3.360 Publikationen ist kein Beweis für das Masernvirus enthalten.
Right. He knew that that was going to happen because he knew that the lower court wouldn't look at the evidence.
Yeah.
And the same thing, I have a good friend who who put up $100,000 for anybody who could show HIV from somebody with AIDS. And I guarantee he still has his $100,000 because there is not one isolated reference of HIV from any person with AIDS. So measles, AIDS, coronavirus. I mean, we're about to do a study, hopefully with Stefan, that shows that there is no virus with people with COVID, and that this process you could literally show that the person has polio or measles or any other RNA virus by doing the same process they're doing. So getting back to your question, so to this person you say, okay, now we've agreed that it's not a virus. Now, interestingly, the first thing I usually say is, since 99.99% of the research has been done on viruses and essentially none on, so what in fact is happening to people, it's hard for me to have the same level of evidence because nobody is looking into it. Having said that, there's very good epidemiological evidence. There's very good mechanistic evidence, which isn't proof, I would admit, that says that it's a combination of, fear and electromagnetic millimeter wave poisoning and glyphosate and maybe air pollution and a bunch of things. So I've said all along, let's do proper research, isolate these factors, and then I'll be able to tell you what happened. In the meantime, the difference with me is I don't think it's a bad idea to lessen your exposure or eliminate, to say, cyanide or air pollution or toxic fear or electromagnetic field poisoning, which I know denatures the structure of the water in your tissues. I already know that. And so there's good reason to think this may not be good for you. And so the worst that can happen with me is I'm wrong, it's not COVID, but it was better for your health anyways. The worst that can happen if you focus on the virus is we destroy the whole world.
Yeah.
So I think I have a better case.
Yeah, definitely. So would people, so you're saying this is where we move into terrain theory, where we're talking about it's the environmental factors that are producing the disease within the body.
And I don't want to even be restrictive in that. It could be the consciousness of the people.
Yes.
It could be living in an environment of fear and hatred and lies and, and every ounce of water is poisoned. Every breath of air, there's toxins in it. They're spraying all kinds of crap and nanoparticles of aluminum in the air. I mean, and you know, there's radiation exposure from all kinds of sources. You know, I don't obviously know if it's even the same thing causing people to get sick in Sri Lanka versus London versus New York. Why does it have to be the same thing? There are certain commonalities and there are certain new things like millimeter waves, which we know interfere with oxygen utilization, which seems to be one of the hallmarks of this situation. My question is, why don't we look into that? Because I don't see the point of living in a world where every ounce of water we drink is full of about 120 different carcinogenic chemicals. Like, why is that something that's controversial to think we should maybe not do that?
Yeah, it shouldn't be. It shouldn't be controversial.
Right. And so what's the worst thing if I'm wrong is we end up healthier because we're not poisoning ourselves with you know, birth control pills in our water or Prozac in our water or fluoride, which is an enzyme inhibitor in our water.
Yeah.
Like, worst thing that can happen is we end up with pure water, and I'm willing to take that risk.
Yeah, exactly. That's how it should be. But I'm guessing the way that they would look at it is that you might be encouraging people to not take the vaccine or not, you know, take the pharma-controlled solutions to everything, which is obviously, well, that's their argument. And then I know that means it's, you know, it's a huge industry, the pharma industrial complex, you know, and so effectively your message is a massive threat to that huge institution, that huge industry that's making billions and billions. That's the reality, isn't it?
Yeah, but I want to clarify one thing, because if anybody asks me, you know, should I take the so-called Mexican beer vaccine? There is no vaccine. A vaccine is defined as something that comes from a virus or bacteria, but let's say a virus. It's an antigen that creates an immunological reaction, and it affects your immunity to that virus and transmission, right? That's what we think of as a vaccine. To condense that, a vaccine comes from a virus, if we're talking about a viral vaccine, and it affects your immune system and it affects transmission. That's what a vaccine is. So here's what Pfizer says about their vaccine, one of the most popular ones. To build an mRNA vaccine, we do not need an actual virus. They say they never had a virus. Moderna says they never had a virus. They never made anything from a virus. They never had a virus to work with. So it doesn't come from a virus. This is a synthetic mRNA injectable drug that is meant to get inserted into your DNA, exactly like genetic engineering. Now, this injectable drug has never been tested for its immunological effects, and it's never been tested for transmission. It's basically an mRNA operating system. Now, again, here's what Pfizer says. The DNA template was generated via a combination of gene synthesis and recombinant DNA technology. It had no relation to any kind of virus. And when people say, well, isn't that conspiracy theory to say that it's a operating system? So here's what Moderna website, one of the biggest injectable drug manufacturers in the world, says. I'm getting this from them. Recognizing the broad potential of mRNA science, we set out to create an mRNA technology platform that functions like an operating system on a computer. It is designed so that it can plug and play interchangeably with different programs. In our case, the program or app is an mRNA drug, the unique mRNA sequence that codes for a protein. So anybody who thinks that the idea that they're injecting an operating system into your body is a conspiracy hoax, apparently Moderna is in on the hoax. Because I got that from reading their website.
Whew, I don't know where to go from that. So when we talk about the mRNA, and they want to create an mRNA, what were their exact words? System, an mRNA operating system within the body.
Yes.
What does that, to a layman, what does that actually mean? What does that look like? Like, what's the effects
the
of
way
that?
it goes is the central dogma of genetics, which like most things in science turn out to be not true, is that DNA makes RNA makes protein. And it's one direction, not RNA never makes DNA, protein never makes RNA, protein never makes DNA.
Right.
Now, we found out with HIV that, and retroviruses, they're so-called retroviruses because they reverse that and RNA makes DNA. You with me?
Yeah.
And so that changed everything, because then the central dogma, that's what it's called, of genetics is wrong. This is a two-way street. And so that means that if you inject RNA drug into a person, it will be converted into DNA, which will then possibly, depending on what happens, get inserted into your DNA, and then you will forevermore be genetically modified. Meaning you will then have a program in you to make new copies of RNA or protein, which you never would have made before. That's the definition of genetic engineering. Or, as I like to say, the only good thing about using this drug is then you don't have to worry about eating GMO food anymore because you're already GMO'd.
Wow, okay.
Now, if you think eating GMO food is bad for you, imagine what happens if you become GMO. And the answer is, so if you say, Tom, what happens? I have no idea. Neither does anybody else. Except you can then be programmed, as they say, so that we can give you uploads and downloads and maybe put your bank account on this so that you will be then essentially a genetic program which will be interfaced with the 5G computer network, and then you can have a better life. Except if you don't like it, you won't have a bank account. And this better life means you can shop at certain stores like Amazon. And if you don't want to shop at Amazon, too bad, because that's all your computer program works.
And that's even been
proved. Be good for Amazon.
It's good for Amazon.
What?
Well, I was going to say that you've even seen this, that you can actually look at the patent for this. They've actually shown how this works with like a crypto device, which harnesses human output, human energy, human behavior, and converts it into sort of a crypto device or cryptocurrency or whatever they want to call it. But that's actually on patent scope. People can have a look at that. So this isn't hidden information. It's no conspiracy. It's all out in the open, and it's done by Microsoft. It's all listed on the site, on the patent website. So there's nothing buried here. This isn't imagination at all.
No, this is I just got that from what they told me.
Yeah. So can I just ask a couple of quick fire questions just before we finish up here then, Tom? When you're getting vaccinated, what's actually going in you, and why is it that does stop people from getting a disease then?
It depends on the vaccine, but it basically substitutes a worse poison so that you can't go through a maturation process. In other words, something like measles is it's like a snake shedding its skin. So if you take something from that process and attach it to worse poisons, you won't be able to go through that, and you'll end up with a worse situation. And that's why it appears like you get less measles, but what you really get is more sick people.
Right, I'm still trying to get my head around that. So it actually does stop you from getting measles, the disease, but it will create more secondary issues, more secondary diseases.
You have to see the measles is the pus getting out the splinter.
Right.
You could take something from the pus and stop you from making pus. And then you're just left with a bunch of splinters. That's how it works, essentially.
Right, okay.
And what about the argument that why is it then that medicines have worked? You know, for instance, HIV, when the advent of the HIV medicines was, you know, people were now living like long lives with, you know, and they're HIV positive, but they're living very long lives. Why is that? Because
HIV positive means nothing.
right.
And that's a very complicated answer, which we don't want to get into right now, but it has to do, those are not antiviral drugs, those are chemotherapy.
Right.
And so basically they're also antifungal drugs. So they're basically killing off fungus, calling it antiviral, and then certain things happen, and sometimes people live longer with a lot of toxicity.
Okay, Tom, we did an hour there. I'm sure we could have gone into way more. Was there anything else you wanted to add to tie the loop on anything?
Not right now. I think we're good. And I hope I accomplished my mission, which is
you did.
Nobody will ever be able to snow you again.
Yeah, that's the idea. Yeah.
And if you talk to anybody who says, "Oh no, I have this paper," so the important thing is you don't ask somebody, "Have you isolated the virus?
Right.
Have you characterized it? Have you shown it caused disease?" Because they'll say yes. The question is, explain to me in very clear way, like hopefully I did.
Yeah.
How you did this. Right? I mean, I tried to be as clear and transparent as I could so that anybody who's interested can understand it.
Yeah, you did. Yeah, and that'll be the way forward. That I'll ask people about that. Yeah.
Great.
Yeah, fantastic. You did a really good job, and I thank you for bringing your props along as well, and you know, the knives and forks and everything else. I really appreciate that because it's good. It's good visually to see it all in plain sight. You know, the separation process, and it's
how human beings think about the world.
Yeah, exactly. Yeah.
And don't be confused because it's something that's complicated and you have to have a white coat to understand it. It's still a thing. Things need to be separated to know what they do.
Yeah, absolutely. Yeah, and they haven't done that. So that's the bottom line. That's the bottom line. Tom, thank you so much for coming on. And yeah, you know, this is an important message. It's a controversial one for a lot of people because they're so entrenched within that current way of thinking and operating. But the only way we move forward as a species, as a civilization, is to question the norms and to find the anomalies and to create these massive, you know, paradigm shifts. And it really, it's not even complex, like you said. It's actually just common sense. It's actually more simple than what we're being told, which is the kind of the irony of it all. It's like it's simplified what you're saying to the point where it's crazy. We're actually living in the abstraction, whereas you're just showing the natural. I think is the kind of what we're demonstrating here. So Tom, you know, all that's left to be said is you know, continue to show your courage and your bravery and speak out and speak up and you know that you're doing the right thing. And I'm going to continue to do the same, broadcast people's messages who I believe in, who I think are doing some really good work. So on that note, Tom, thanks a lot, and we'll speak to you soon.
Okay, bye-bye.
Cheers now.